Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Tēnā koutou katoa listeners. Welcome to this episode where we're talking about artificial intelligence in plant science. You must be thinking, what's the point of an artificial intelligence or AI plant if you can't eat its fruit, if you can't watch it grow, if you can't breathe in the oxygen? But maybe it's just me that thinks that way. So here's a plot twist. Today's guest is Sarah Philp-Wright, a researcher based here at Tamaki Makaurau, Auckland. Welcome, Sarah.
Hi, Jay. Thanks so much for having me. I'm really excited to be here.
So, Sarah, how'd you get started with PFR and working with AI plants?
So it all started a couple of years ago when I was a summer student here at Plant and Food Research in Mount Albert with the plant development team, who's led by Dr. Kim Snowden. So I joined the team and got to learn all about the work that they did, and I just fell in love and I haven't left. So with them, I did my BSc honours, which I recently finished at the start of this year, looking into those AI-designed plants. I'm also based as well at the University of Auckland, so I was co-supervised at the university. And now since I finished my honours, I work here as a researcher at PFR, as well as a researcher and technician at the University of Auckland and as a tutor, so teaching an undergraduate bio courses. For my honours, I was really fortunate to be awarded a scholarship from the New Zealand Fruit Growers Charitable Trust and Horticulture New Zealand. So they provided me some funding to support my research, as well as opportunities to actually get involved with New Zealand's horticultural sector as well. So I would highly advise people to apply for the scholarship because it's an awesome opportunity.
Okay, excellent. That's something interesting you said. You said AI designed plants as opposed to what I said. I said AI plants. So maybe that's not significant, but tell me a little bit more about it.
So a little bit about what my research focused on. So we're looking at how we can use AI to create new proteins or new inputs or even new genes that we can add into plants to alter their growth and their development. So we're looking at those new inputs and how we can improve the plants we have already.
Right. So you're doing some studies in, let's say, a computer and then trying to translate that research into an actual plant. So there are real plants involved. Interesting. So there is something to be eaten after all. Great. So let's talk about what you're specifically doing. What sort of parts of the plant are you interested in?
So as part of our team, we're really interested in plant shape and architecture. So that's its growth habit, how it grows, and especially its branches. So we're focusing a lot on branching and the regulation of branching by hormones, such as strigolactones. So we know this family of hormones that regulate growth and development just like hormones do in humans, but they have multiple uses that they affect plant height, branching, as well as different leaf traits, all sorts of things. So we're looking at how we can design proteins using AI to alter that hormone signaling, so alter strigolactones in the plants, so we can change their downstream effects and alter plant shape and architecture.
Why is that important? Why is plant shape or architecture really important to us?
So plant shape is actually one of those sort of key economically important traits. It has implications for things like yield as well as productivity and management. Because plant shape, it determines things like how big the plant is, how many branches, and of course how much fruit you can get on a branch. So if you think, for example, like management, if your plant is really bushy and really large, it can be difficult to prune. There's more labor costs and time trying to manage it. So if we could make that plant smaller without sacrificing the yield, make it easier to manage as well as fit more plants in a given area, that increases our productivity.
Okay, so I'm imagining something like dwarf varieties of, let's say, fruit trees. Is it that sort of thing?
Exactly. So we're looking almost like an alternative to dwarf varieties, as especially a lot of crops that we use at the moment, dwarf varieties are either created through breeding programs or grafting onto dwarf fruit stocks. But what if we could find ways using genetic technologies and biotech to increase the speed and have a new way to develop those dwarf cultivars?
Cool. So then how is AI involved in this process?
With recent advances in AI technology, They now allow us to do all sorts of things, but in particular design proteins, compounds, small molecules, including ones that have never existed before in nature. So what we can do, if we think of a target, so in our case, we looked at a hormone receptor, and that is our target protein. We then thought about how we could design a protein that could stick to it physically or somehow alter or inhibit its function so that the hormone could no longer be received and it would stop signaling downstream. Once we chose our target, it's all in the genius of a structural biologist in our team. So I'm not doing this all alone. Science is often more than one person.
A team sport, yeah.
Exactly. So we're really fortunate to have a wonderful structural biologist on our team, Cyril Hamior. So as I talked about recent advancements in AI, and we actually saw that was published, was this new software called RFdiffusion that now allowed us to design proteins de novo. So that's completely from scratch, that by choosing a target or feeding in certain conditions, it would generate new protein structures, some of which don't exist before in nature, so they're completely novel. So we thought, how could we use that technology, that design process, to solve the problems that we have in plant science and things like altering plant architecture? So we decided to go for the hormone receptor that picks up strigolactones in plants. And how the software works is you can give the software certain conditions. So for example, certain places on a protein you want it to bind to, or you want it to have a certain functional group, which is an area on the protein that may carry out a certain function or do something specifically. So a little bit of top secret knowledge. Well, our structural biologist chose the conditions and put the instructions into the software about what sort of protein structure we wanted it to create in the end. And then we just let the AI software run, and then it spits out a certain number of candidates, completely randomly generated. We just specify how many candidates, and it gives us a whole random bunch. So from there, it gives us protein structure. We then use other software to assign it a sequence. We can put that sequence into plants and they can make it themselves, or we can get the protein synthesized as a gene. And then after that, using the knowledge of our structural biologist, he then filtered down the candidates. So we had our top 10 ones that we thought were most likely to bind our target, so that strigolactone hormone receptor in plants. And that's where my job came in, that once we had our 10 candidates, I was going to test them to see which ones actually worked in real life. We had predictions. We narrowed them down using in silico computational techniques, but just because they work on the computer, just because the AI says it will work, doesn't mean it necessarily works in real life.
All right, perfect. So you're turning artificial intelligence into actual intelligence.
Exactly.
I like that. I just want to take a step back. So we talked about traditional ways to achieve things like dwarfing. Do they give you any clues about that original protein target you're looking to find? Or is this something that you learn from model plants? Where do you get that original protein to then look for binding partners using what you call RFdiffusion?
So it's a lot of the stuff that you've already mentioned. Based on the current knowledge we have, the research that my team have done, as I mentioned, we know that when that hormone receptor is knocked out in plants, it results in plants that are shorter with more branches, so it's more compact. So we thought if we could use a new method to emulate that same effect, but with this AI design method or these AI design proteins, they may have some advantages over current existing methods that we use now. We thought, how can we get there using a different way? So we build on the knowledge that we've already had through years of research, and we know some of the factors that regulate branching in plants.
Okay, and so those are the ones you target through RFdiffusion.
Yes, so we chose the receptor because we've done lots of studies that show when it's knocked out, when it's not functional, how that affects plant traits further down the line.
Right, and so now you're in the journey using AI to try and figure out partners that can interact with it and get desired outcomes based on what's already known about that gene or that protein's function.
Yeah. So based on the research we have, we choose our target and we kind of have an idea of if we change or alter how that target functions, then we can change traits in the plants. So we're trying to get right from designing some sort of protein, adding it to a target to have some big downstream effect.
This sounds absolutely wonderful and crazy, like sci-fi level. So RF diffusion, is that really key? What is RF diffusion?
So RFdiffusion is a pretty cool software, if I do say so myself, that was first published in 2023 by the Baker Group, I think in the US. And that is a software, it's a de novo protein design software that, as I mentioned, allows you to design proteins, scaffolds, all sorts of different things like binding partners, depending on what conditions you give it. The basic premises is that they designed this software, they trained it on a database full of known protein structures. And then they scramble it up and then they give it back to the software and it has to learn how to unscramble it and how to build a protein from that. If you feed in certain criteria or conditions, say for example, you want this certain shape or you want this protein to be able to stick to this enzyme or inhibit this activity, because it's learned from all the knowledge that we have about protein structure and the databases, it can then spit out something based on the training data that it thinks will carry out the functions or meet the criteria that you've specified. The caveat is that although it should work online on these computer based on in silico testing, it doesn't always necessarily translate into that real life effect. So you need to do more experimental validation, which is also always important.
So the Sarah Philp-Wrights of this world are absolutely essential to testing whether it's real or not.
I think that's a testament to AI, that as we use it more and more, it's often not just a one and done. You don't just put something into ChatGPT, take it out and, there we go. It's already all done. But rather, using AI is a really useful tool that still, I think, requires human oversight as well as editing and refining. So it's great for making a process more efficient, for speeding it up, and even creating novel designs like these novel proteins that don't exist before in nature that human researchers may not have thought to create. It's a tool, it's a step, but it'll always require, I think, some human touch to perfect it or to improve it.
Okay. So you talked about RF diffusion coming from the Baker Lab. So that's David Baker from the 2024 Nobel Prize in Chemistry fame, shared with John Jumper and Dimas Hassabis, who together launched RoseTTAFold and AlphaFold that listeners may be aware of. So how is RF diffusion different to RoseTTAFold and AlphaFold, and how are you using it differently to what people may be already familiar with?
So I talk about RF diffusion, RF. That RF actually stands for RoseTTAFold. So the difference between RoseTTAFold and RoseTTAFold diffusion, RF diffusion, is that RoseTTAFold, you have a protein sequence, so that sequence of amino acids that you know will fold into a protein, but you put it into RoseTTAFold and ask it to predict what you think the 3D protein structure will look like. Whereas with RF diffusion, you have certain constraints or conditions. You think about what you want a protein to do, and you tell RF diffusion to make a 3D protein structure that should theoretically fulfill those functions.
Okay, so you've got RF diffusion, you've designed some hypothetical proteins or protein structures, you've then gone on to synthesize the genes associated with those predicted proteins. What next?
Then it was time to put those proteins or the genes that we've gotten that encode those proteins into real-life organisms to see if they have actually any effect at all. Do they do what we intended? Do they do nothing? We didn't have much of an idea. Based on our in silico, our computational validation, they had a high likelihood of binding our target receptor, but just because it says that on the computer doesn't mean it will do so in a plant or in another organism.
Okay, so you have to get it into the plant somehow. Is it plants that you're testing them in?
So first up, because we had 10 candidates and actually testing them in plants can be quite laborious. It's a lot of work, a lot of effort, and it can take a lot of fine tuning. We wanted to screen those candidates initially by using a yeast hybrid screen. So we actually transformed those genes into yeast, and we use this yeast system that's already been established to see how well that our candidate proteins could bind and interact with our receptor.
Okay, so you have this screening system to narrow down your candidates. How many did you end up with after that?
So from the 10 that we had, surprisingly, we actually had 4 candidates that showed interaction in yeast, because how it works is that we put our designed protein into yeast, and then we put our receptor into another type of yeast, and we put them together. And if the 2 proteins, our receptor and our designed one, interact, the yeast grows.
Okay.
So we saw definite yeast growth with 3 of our candidates immediately during the test that we did. And then we saw another candidate took a bit longer to get there.
Okay. So, 4 is pretty impressive. I've done yeast 2 hybrid type work before, and 4 is just incredible. I guess the AI did prove to be a very, very powerful tool in that aspect.
We were absolutely stoked that we actually had something that interacted at all. But interestingly enough, I think it highlights another difference, that we designed it to target the hormone receptor in one species, in petunia, which you may know from the garden centre or in your garden.
Oh, yeah, of course.
We actually tested it with that strigolactin receptor from a couple of different species. So petunia and another species, a common model plant, which you probably have heard of as Arabidopsis.
Our old friend.
Exactly. Yep. It's everywhere. So although we designed the proteins to interact and bind with the petunia receptor, actually our 2 of those 4 candidates showed evidence they only interacted with the receptor in Arabidopsis. So they were designed to hit petunia, but they only actually interacted with Arabidopsis. Whereas 1 candidate did work with petunia only, and then our 4th candidate interacted with both in yeast. So it's an interesting difference that although they were predicted to all bind with the petunia receptor, 2 did and 2 didn't, and 2 did with another species.
Okay. Interesting. So you've got like a mixture of results. Obviously the success rate, you can't argue with that. So now you've got these candidates. They do interact with the receptor or receptors that you think that they should be interacting with. What do you do then?
So just because they interact in yeasts are very different to plants. So they may not have the same or any activity in plants. So my next step was to transform the genes, so transform those 4 proteins into the plant so they would express them themselves, and then observe how the plant developed and see if there were any changes in its growth in any desired traits.
So in this case, it was architecture. So what did you see? Did you see any changes?
I put them into Arabidopsis because it's a great model plant to test it. And from that, we noticed that actually the plants were generally smaller. They had smaller rosette growth, plants that carried the binders, compared to our wild type plants that had no binder.
Okay, so is that what you expected?
We grew it alongside a receptor knockout plant. So that is kind of our positive control. If you think it's our target of what we'd expect, if this protein is actually inhibiting the strigolactin receptor, we should see traits similar to this receptor knockout, where it's got a non-functional strigolactin receptor in it. All 4 lines that carried a protein, they had that small rosette size, so smaller growth, which was similar to our positive, our receptor knockout. So that's what we expected. So it suggests that maybe it does have some early effect, that it is inhibiting the receptor in early development.
Would you say that it's a dwarfing phenotype then, as you expected?
But unfortunately, once the plants matured and they got older, there was no significant difference in the height of plants that had the proteins in them versus plants that didn't. And there was no difference in things like flowering, which can influence yield. But we did see that plants that carried one protein appeared to actually have reduced branches, so fewer branches than all the other lines and all the other plants that didn't have the protein.
Okay, so you saw some phenotypes coming through. So what do you do going forward? How do you tackle this?
There are lots of different steps that we could take. One is seeing if we can change how the protein is expressed in plants to see if we can modulate the strength of its effects. See if we can dial up its expression so there's more protein in the plant and maybe we'll get a stronger effect. as well as looking to see if we can put that protein in and make it expressed in certain areas of the plant to get variable effects. But also things like we can go back to the drawing board. Maybe the structures of these proteins themselves need to be modified slightly to improve how they work. See if we can alter the protein to alter their effects on plant traits. And of course, we need a lot more testing, other lines of evidence to show that they do interact and look at how they interact in plants. Because I've gone from kind of step 0 is screening candidates to get 4 proteins that we think might work to then throwing them into the plant and look, it has some effects maybe on early development, which are similar to what we would expect to happen if the receptor was knocked out. But I haven't mentioned that reduced branching is actually the opposite of what was expected when the receptor is non-functional. So it leaves a huge gap of what actually is going on inside the plant. What is the protein up to? How does it work? Is it binding and inhibiting that receptor as we thought? What are the downstream effects? So there's lots more research and experiments that we can do to figure out how those proteins work in plants.
Well, it sounds really interesting. It sounds like actually just the beginning of the story. I would have imagined AI would just give you the answers. You know, you can cheat on tests, so why not just cheat and give us the plant? But obviously, a lot more testing is needed. The go-to phrase in science, of course, is a lot more testing is needed. So where would you like to see this bespoke sort of protein design technology progressing in the future?
I think ultimately what we're looking at with these proteins, in the case of plants and plant shape and architecture, is either the basis for new growth regulators or a new cultivar. So there's the option that once we get some proteins that are working, which have the effects or the desired effects, desired strengths and things like that, we could look potentially at introducing those into plants either through exogenous external methods like topical application. If you think peptides or sprays, what if you could apply that protein on the outside of the plant to alter how it grows? But that in itself has a lot of hurdles to try and perfect. Just putting something on the outside of a plant and magically changing how it grows. But the other option is, depending on what regulations look like, especially the legislation, is actually using transgenic modification approaches and introducing those proteins into plants so that they express them themselves. And then it could ultimately result in new dwarf cultivars or, more excitingly, more novel plant shapes, not just dwarf, but intermediates. And you could have more flexible changes or more subtle changes in plant shape, height, branching, because if we can put those proteins into plants, either transforming them in there or through topical application, but if we can change how much protein is in there and that changes the strength of the change in the plant, if we could introduce those proteins into certain aspects or at certain times in certain tissues, what if we wanted to control how big a plant is when they're young? What if you think of plants in a nursery and you want, for easier management, you want them to be smaller, more compact. Mm-hmm. But later, if you stopped expressing those proteins and let them grow out in their original wild growth pattern later, once they'd been spread out in the field. So it's things like, how can we use these proteins to alter plants to improve their growing for lots of applications in horticulture and agriculture, but particularly controlled environment agriculture, urban agriculture and growing, things like covered cropping and indoor environments, especially as populations are growing in the future, that resources are running out, thinking about how we can improve the productivity of our plants and making them more compact, maximizing their yield.
You're talking about crop intensification, right? So small footprint, high yield, high production.
Yes.
You covered something interesting. You said transgenic plants, or you can spray them with peptides. I was just thinking about whether maybe there's a feasibility in maybe doing some gene editing. So I imagine, you use AI to predict some radical protein that interacts with your partner. But, these proteins exist in plants. They have natural partners. would you envision maybe having that partner, the natural partner, being modified slightly in some way to change interactions with its native partner? Would that be an avenue that could be explored? It's not transgenic, it's gene editing.
Do you mean gene editing the receptor?
A receptor to change it and its partner's interaction, or changing the partner to optimize or reduce its interaction with the receptor. Something like that?
Yeah, so gene editing is already being used in this space, even with the target that we've chosen,
Right.
that we're already able to gene edit it and knock out its function to have those effects. But this use of AI-designed novel proteins is more like another tool in the toolbox. So it's not completely replacing all other methods, but it's adding another avenue that we can use in partnership with lots of other methods to control plant growth and plant development. But the difference between them, and sometimes the advantage of having our AI-designed proteins and introducing them into plants, sometimes it's easier to add something in rather than trying to gene edit and CRISPR knock out a gene or multiple copies in a plant. But also our proteins may offer some advantage in being multi-species. So if we can find an AI-designed protein that has similar effects or even different effects in multiple different species, then we can release that same protein, we can transform it into different species to control how several plants grow, rather than gene editing often needs to be more species specific because genes vary a lot more between species, and certain species have multiple copies of that same gene where they'd all have to be edited to get the effect that you need.
Yeah, I can see that, especially for the peptide-mediated manipulation. You generate a peptide spray that would work on a number of plants and have a significant desired outcome from that spray, from that perspective. It's very exciting. So AI is a new tool in plant science. What are your thoughts on how AI can help science in the future?
I think AI has enormous potential to help several different aspects of science. So we already see it's being increasingly used and applied in settings such as healthcare and medicine, drug development, designing new antibodies, vaccines. I recently saw an awesome paper using RFdiffusion where they designed snake antivenom.
Cool.
So already we actually incorporate a lot of AI into even plant science because of things like helping us to process data, choose new breeding targets, streamline our breeding programs, and all sorts of things. We can use this AI design method to design proteins for lots of applications for plants, humans, animals, the whole lot, but even enhance plant systems, not just hormone signaling like our group are targeting here, but if we could improve the efficiency of different enzymes in plants. So we designed these proteins to inhibit and block the function of this receptor. But in the same way, you could design proteins that actually enhance how it works. You could strengthen that signaling and have more extreme responses in the plant, as well as you could also just replace that receptor. Maybe not this receptor, but in general for the uses of AI in science, you can replace enzymes to make them more efficient, more robust, and improve lots of different aspects of plant systems. As you've talked about, different protein interactions. So we can already gene edit a lot of things, but using these AI-designed proteins to target other proteins, to change all sorts of interactions in plants, really the opportunities are endless.
Yeah, it sounds it's incredible. And I suppose the more you play around with it, the more you use it, the more you learn, and the better we get at trying to deploy applications for AI-enabled science. Very excited for the future. And it's great to have you on board as a partner in this journey. Very excited for your involvement. I'll keep an eye on your science, keep an eye on your publications, and maybe we can have a chat in the future where you can tell me the exciting new avenues you've discovered using AI.
Of course. I'm always interested in reaching out, talking a bit more, but also learning. There's always so much going on, especially in the field of AI research and AI development and protein development and plant science. So it's always nice to keep a finger on the pulse and see what's happening.
Thanks, Sarah.
Thank you.
And thank you, listeners, for joining us for this episode of Scigest. If you liked what you heard, please recommend it to your friends. Word of mouth is the best way to grow. If you didn't like it, please recommend it to your enemies, because word of mouth is still the best way to grow. Please rate and review us on your favorite podcast platform. That helps us out a lot. And catch you on the next episode of Scigest.